Okay, guys. Are you? We're gonna go ahead and move forward with the next part of the morning. We are super excited to have Bob Kuhn back again. Bob is from the UCSC browser. He's an assistant director there. Associate director there, with a particular interest in clinical genetics. Last year, his workshop was one of the most well enjoyed parts of the conference, and people were asking for more of that, especially because when you leave here, you will be able to continue to take advantage of the tools that Bob is going to walk us through today. So with that, I'll welcome you.
Well, thank you so much for the nice introduction. I didn't know it was one of the most enjoyed. I enjoyed it. I knew that. So thanks, everybody, and welcome. And thanks to the organizers for having me back. It's a pleasure to come visit. I learned so much yesterday. I was really wishing I had been here for the earlier days of the course, too. But there's only so much time I can sit and just listen, you know. Did everybody get one of the little mini handouts? Okay, so these are our mini handouts. You know how conservative we are in California, so we wanted to conserve paper. So I made it just big enough for somebody like I can read. You know, the older you get, the shorter your arm gets. And so the bigger the print has to be. Okay, so this is another slide with some redwoods on it. You didn't expect to see redwoods in 2 talks in a row, I'm sure. But so my plan today is to spend a little time with some introductory slides to kind of set the stage, tell you why the browser's cool, and then we'll spend some time playing with the browser and show you some of the things that you can do with it. We're not going to have time to go deeply into many of the features. But as you know, the first step in using any tool is knowing what the tool's good for. And then some things will be useful to certain people and other things for other people. And then you'll have a chance to dig more deeply into it. So I'm going to start with disclosures. I get some royalties when we sell licenses to commercial companies. You can download our code and run it on your machine for free if you're at a hospital or an academic institution. You're supposed to pay for it if you're at a company. And we got like 1 million lines of code and 300 FTE years in the browser. And so that's the opportunity for commercial outfits to pay back the government for all of the funding. And we have a contract with Regeneron to do some work, most of which winds up back in the browser, but some things that they want prioritized, they are a little bit of a funding stream for us as well. Our main funding is from NHGRI. Can you see that okay? Because there's like a light on the screen, which is a little weird. But I know I don't want that being totally in the dark. But and we also have some money. The next 2 are California initiatives, and the bottom 2 are private. And they fund our project to various degrees. NHGRI is our primary funder. The team consists of David Hausler, who's a professor at UCSC, and it's under his umbrella that we work, but we're kind of like a software company inside UCSC. And Jim wrote the original code, and he's the team leader in PI, but Ann does all the management, and I do all the outreach because Jim's a coder. It's what he does well. That's why the browser is really good. And he continues to contribute to the browser on the engineering end. We have a bunch of great engineers who have a background at a lot of companies that you've heard of and universities you've heard of and so forth. But the QA team is really unusual for an academic research project, software project, because most teams are built by, or most software projects are built by a grad student or a postdoc who moves on, and then there's no continuity and no support. But we're in our 20th year now, and the browser owes some of that to the QA team because they're really good at breaking things, and they break it before you get to see it broken. And they pound on it really hard with manual testing and with scripts and so forth, and they test our data and check it out before we release it. So I want to give a shout out to them, and to Jorge, Eric, and Haifeng for keeping all of our back end running, all of our clusters and all of our computational stuff. And the website's getting around 1 million hits a day now, and Jorge, Eric, and Haifeng are the reason why it doesn't break. We also have mirrors in Germany and Japan if you're over there and you want to get a little faster experience, you can use one of our mirrors there. Or if you have colleagues over there, they're automatically redirected the first time they visit the browser. So here's where we are. We're on the central coast of California, across the bay from Monterey and across the mountain from Silicon Valley, which is both a source and a sink for engineers. Some of our engineers have experience over there, and some of our engineers are experiencing over there right now because we lost them. We can't afford to compete with Google for the cost of an engineer. We're supposed to call ourselves UC Santa Cruz, so you don't confuse us with all of these other things. Some of them are UCs and some of them aren't. But and here we are in the Redwoods on campus. There's Jim, the team leader. I'll talk about some features built by Galt Barber, Angie Hinrichs, Kate Rosenblum, and Max Heusler, and Brian Lee. Where's Brian? Right over here. He runs the QA team, and so his people are the reason why the browser doesn't break very often. Oh yeah, and this is to remind me to acknowledge Emily. Is she in the room? There she is. I want to give Emily a little thank you, a little acknowledgement for inviting me and having me back. And your own UCSC Genomics coffee mug. And if I don't put the slide in there, I forget to acknowledge you and thank you for having me here. And then it's in my suitcase and I wanted to bring it at home, so I didn't want to do that. So thank you, Emily, for that. So you already have found your little mini handout. So I want to tell you that a way to dig more deeply into some of the features is the YouTube training channel. There's a short link to it. You can also get to it on our training page. There's a link to it. And if you think that what I've given you today is the tip of an iceberg and you'd like to see the rest of the iceberg, you can contact us via our training page, and I will come and give a more lengthy workshop. I give 1-day and 2-day workshops. We're relatively affordable because my salary is covered by NHGRI. We ask for logistical support and little else. So you can contact us there, and I can tell all of your colleagues all about the Genome browser as well, and I'm happy to do that. There are the links that are on your mini handout. And what is the Genome browser? It's a display engine for genomic annotations. It's essentially the coordinate system of the genome. Many genomes, actually. We have 120 animals. You can load anything that can be mapped to a coordinate onto it. Where are the genes? Where are the enhancers? Where are the histone modification sites? Where are the DNA methylation sites? And so forth. If they can be measured, they can be mapped to an oligo, they can be mapped to the genome. We can draw a little box and say, here's where it is. And then you can click through to it and read the details. We have a consistent interface across all genomes, so you don't have to relearn navigation when you change from human to mouse to fly to worm and so forth. But I like to think of it as a more dynamic thing. It's an inquiry-driven tool to give you answers in real time to your questions. You don't have to go all over the internet, or used to have to go all over campus to go to find the books in the library and look stuff up, or go to the bench and spend a year figuring it out. Oh, I wonder if that gene's expressed in mouse. You know, you just shrug and go back to work. Now you can click around and find out in a few minutes whether it is or not and what the mismatches are and so forth. So you can get answers that can drive your inquiry in real time as you have the questions. So it's built out of data that come from the outside world. Even the underlying genomes come from the outside world, and we do a lot of computation on them, especially our comparative genomics pipelines. And they ultimately then wind up an annotation, something like a methylation site that's been mapped, can be mapped to the genome, and the coordinates of the mapping can go into a MySQL table. We put all the mRNAs from GenBank in there. So all of the rat RNAs that align to human are in a table, and all the human RNAs are in a table. And so the coordinates are just our coordinates, so we can draw a little box, and the actual sequences wind up in a flat file that's accessible via a click-through if you need it. So everything's condensed, and it's already pre-aligned, and so you can get to the answer really quickly. We can draw a picture with 100 RNAs in a couple of seconds, and you can see all of the RNAs on a big chunk of the genome very quickly. So we're optimized for speed, and the whole code runs in the C programming language, which gives us really good access to memory management and high-speed compute. So up here, the CGIs, Common Gateway Interface, those are the programs that convert a click on the browser into a database query. and a new picture on your browser. And so that all happens behind the scenes, and we have a lot of different CGIs that do different things. One of them gives us a details page when you do click into an RNA or a histone modification site, and then you can get access to the original data back at the place where the data came from. So we can click through and we can give you the original information. So we don't try to pull in all the data in the world, but we try to get pointers to the most important data so that you can find it. We don't suck in all of OMIM, but we do have pointers to OMIM, so when you're on a gene, you can get to OMIM very quickly. You can get to a specific allelic variant in OMIM very quickly and read about the family that was responsible for that particular amino acid substitution and that change. And so we also have the capability for you to load your own data into the browser. I've seen a bunch of pictures of IGV. They seem to have captured the right off the sequencer space. So a lot of people get the sequences off of their sequencer, put it into IGV. Your BAM files and your VCF files are also loadable into the Genome browser where they're visualizable and also intersectable. So you can intersect them with all of our other data. So you can say, okay, here are all my variants. Which ones of them are also common in the genome? And so you can get a visual view of the ones that are common or the ones that are left over when you subtract the common ones. You can see the ones that are left. And then you can go from gene to gene to gene and see all of the other information you might be interested in. Data that are really large or too complex belong in data hubs because they're too big to upload. BAM files and VCF files are both too big to upload as a static process because it takes forever to upload and it times out. And so in a case like that, the file needs to go on a server so that our server can talk to your server. And instead of loading the whole file, we pull out just the slice of the file that's relevant for the picture you're looking at. And so that makes it a very quick transaction. And so if you're interested in data hubs, we have a lot of documentation on that. And it's a little bit of heavy lifting. It's good to get a bioinformatician involved in helping you set that up. And you can also get our team involved. Our mailing list is there to help you over some of the hurdles. We know a lot of our end users aren't Unix comfortable, and getting data into a server isn't trivial for them, but that's why we have a team to help. We have people that answer the mailing list and help you through the process. An alternative to putting the data onto a data hub, making it accessible to the browser, is to download the browser to your desktop. So we have a version called Genome Browser in a Box, which is a single laptop instance. It's kind of big. It's about 10 gigs to download. But once you have it downloaded and installed on your machine, then it sits next to your data, and you don't have to expose your data to the web, which you might not be allowed to anyway if it's not properly de-identified. So then your data are next to the browser, and you can load it directly into the browser. And I almost said go to the beach, but you can go out to the reservoir down by the river and work offline if you like. You would need to have access to UCSC if you wanted to load a fresh data set that you hadn't cached, because in that case, our data are now remote from you, and so the data have to be pulled in from the outside. There's a link on our blue bar. I often forget to mention it. Under downloads, it's called Genome Browser Store. And you can go to the store, and if you're at an academic institution, you can buy it for $0 and do the download. If you're with a profit-making company, you're expected to pay the license. And then you have a copy of the browser, and as best we can, it works just like the online version. So once you're clicking around in it, the activity is the same. The interface is all the same. And so you can load your data into the browser and see it next to ours and intersect it next to ours with the table browser, and using all of the other features. So there's really two kinds of data, our stuff and your stuff, and you can see them together on the browser. And so with respect to loading your own data into the browser, there are a number of different data formats that we support for input into the browser. And so for displaying your data, you can load and see your individual reads, just like you were seeing in the previous talk, all the individual little strips that say, okay, here's where a read is. And you can see where the mismatches are. You get that for free, and I'll show you that in a little bit. You can load a BAM file or a CRAM file into the browser and visualize your data. You can also see coverage, which is essentially taking the reads and counting them up and giving you a number, giving you a curve that says, what's my coverage at this exon, what's my coverage at that exon, and so forth. And so you can get a continuous curve you can load the BAM file and you can get it to show you the coverage, and I'll show you that. Or you can convert it to a wiggle file, which is just a continuously variable file. If you have variants, VCF and PGSNP are input types that you can load data into the browser in these formats. And we also accept HGVS and RSIDs into one of our tools as input. And I'll mention that when I get to it. And that tool is the Variant Annotation Integrator, which is a predictive tool. It lets the genome browser tell you what your variants are going to do at the biochemical and somewhat biological level. It'll tell you if it's a missense or if it's a splice variant change or a frame shift. And it'll also allow you to do a little bit of prediction using SIFT and polyphen or mutation taster and a few of the other predictors that help determine whether or not it's likely to be biologically pathogenic or benign in a biological context. But we'll put predictive in quotes because it's algorithmic and it gets things wrong. And I have a nice example I intend to show you later where SIFT and polyphen don't agree. One calls it pathogenic and the other calls it benign, which is cool to know. If the algorithms don't agree, you want to know that. So you don't want to slavishly follow one and not know that others have different approaches and have different opinions about that. And in assistance of the data, the predictive function of the browser is we have data tracks that allow you to see copy number variants in single nucleotide polymorphisms or variants, both benign and pathogenic, so that you can use those data yourself and do it in real time, looking at the data and decide for yourself what you think of that. So among the objectives I have today are to expose you to all of this stuff in the short time that we have together, talk a little bit about the differences in the human genome assemblies, talk about how to get around in the browser, how to save your thoughts and share them with somebody else, how to load your own data into the browser with custom tracks, some of our data sets that are relevant, talk a little bit about conservation, showing you the 100 species multi-Z conservation track, use the table browser to pull some coordinates out for genes. I had a good time yesterday with one of you during the talks. We were doing that, pulling coordinates out from a list of genes, using a longer list than I intend to use today. And then we can use those coordinates as input into a function the browser has so you can look at just those regions. If you have a handful of genes, a gene panel, or a pathway you're interested in, you can look at just those regions and ignore the rest. We also have exon-only mode if you're doing transcriptomics or exome sequencing, and you don't care about the rest of the genome. You can look at just the part that's interesting to you using exon-only mode. If we get to it, we'll look at ISPCR and the Variant Annotation Integrator. So the genome itself started, and I want to thank Jay Shanduri for drawing my attention to this by publishing this ad. It was in the Buffalo News in 1997 asking for volunteers. And one of the volunteers that answered this ad became the RP from Roswell Park, the RP11 library, which turned out to be 71% of the final genome. It turned out they had a really good library, and even though all of these different labs were sequencing different chromosomes and having their own libraries, one by one the libraries petered out and they kept finding the same clones and they got no new stuff. You know, the complexity of the library was too small to have a really good coverage. And so they cloned by phone. They called each other up and say, send me your copy of your library. And before you knew it, RP11 was the thing. And it wound up being 2/3 of the reference assembly. So it's really just one chromosome of one guy over 2/3 of the genome. And the other 1/3 is a mix of the rest of those people who were in the de-identified sample. And so the reference is what it is. It's going to have some places, and there are places where there are private SNPs, where that guy had a variant and nobody has seen it since. That's something to keep in mind when you're doing your bioinformatics. Is you see a variant relative to the reference, it could be that the reference is the weird one, not you. You know, all of your samples may have those same variants, and most bioinformatics pipelines now are aware of many of those. We now have 660 million differences in dbSNP that are mapped on a data set that we have on the genome browser, so you can see all of those variants. So that's roughly a third of the genome now has been found to be variable, have a variant in it, at least in one individual somewhere, and we have all of those data on the browser. So the whole question of variation and how does my sample differ from the ref, and how do different samples differ from each other, can be demonstrated by two of the people who put their genomes on the web early on. Watson and Venter, even though they're both white guys, they have only 1.2 million of their variants in common of the 2 to 3 million that they have relative to the reference. So you've heard yesterday somebody said 5 million differences. Any sample from the reference or any two people will differ from each other by that many. But there's going to be some overlap where you differ from the reference and somebody else also differs from the reference at that place. And what the browser does for you, it lets you see those data mapped against the reference. And so here, whoops, not yet. And so here's Watson and Venter in our data set. We have about 120, I guess, individual genomes. Back when it was not so common to have individual genomes on the browser, we started, I mean, in the public, we put them on the browser. Now we don't add any new ones because they're coming out by the thousands. But you can load yours in there as well as a custom track and see yours. And if you want to intersect it with Watson or Venter, you can use the table browser to do that. I use the table browser to intersect Watson with Venter, and you can see there are places where they have variants in common, where also here's a place where Watson has variants Venter doesn't, and here's a couple of Venter variants that Watson doesn't. And you can use the table browser to do that. You can also use the table browser, for example, to sum them and say, what's the total variation between two people? For example, what's the total variation that a mother and a father might have contributed to the child? So if you have a trio, you can add the mother to the father and see the data on the browser by loading in mother plus father, and that's all the variation that would have been heritable to the child. Then you can use that as the raw material for another intersection where you could subtract that from the child, leaving behind a data set that showed the de novo variants. And so now you have data sets on the browser that you can look at, and you can look at in detail each one of those variants. And so what the browser does is let you load all of them in together. Here's that 1 part per thousand, because it's roughly 3 million variants out of 3 billion. So basically you're throwing away most of your data when you get it down to the remaining variants, but you still have a couple million or 3 million things to dig through. This is a look at all of the releases of dbSNP through the years, where these are different variant releases. And at a certain point, we discovered that people were subtracting dbSNP from their samples under the mistaken impression that dbSNP was polymorphic. That P means polymorphism, which implies to a true geneticist that it's 1% in the population and segregating and so forth. But dbSNP also takes anything that somebody puts in it. And as time has gone by, the number of SNPs has gone way up, but the number of 1%ers has not. So we pulled out a data set, the common SNPs, back at the early days of HT19. This common SNPs track hasn't increased very much through the years, because by then we'd already had a few hundred genomes sequenced, and you're already going to find all the 1%ers in the first few hundred. The chances of finding any more of them is very small once you've done a few hundred genomes. And so if you're going to subtract the common ones out, you should be using the common SNP track. So we put that into a separate data set so people would find it. But it shows you the challenge of handling all the data as they continue to come through. The most recent release is more than 600 million variants. So another type of variation that's relevant, especially in this room, is variation with phenotypic significance. I made this particular slide about 5 years ago when the total number of variants was quite a bit smaller than it is now. But the point I want to make about it is that no matter which database you use for your phenotypically significant variants, you're going to have some variants that are known but just not in that database. And so this is an intersection, a Venn diagram that shows all of the variation in the 3 biggest variant databases at that time. And each of them has some that the others don't, and they share some with each other in various combinations. And in fact, only of the 5 databases, only 2,200 are common to all of them. So the chance of having a database at random and a variant at random and having the variant be in the database are not high unless you look at all of them. And so what the genome browser does for you, whoops, wrong way. What the genome browser does for you is lets you see all of them at the same time on the screen because we have all of those variation databases. And some of these variants will be in multiple databases, some will be in just one, but you can see them all, you can click into them, and then you can go read the story about the individual variant. And that's one of the values we have, we add to these databases. There's a caveat that if it's in more than one database, doesn't mean that it's been seen more than once, because they might have gotten it from the same original source. It might have come from the same literature, from the same lab. And so the value of being able to click into the original record and see where it came from is pretty high, so that you might not be, you don't want to be fooled into thinking that it's been seen multiple times and therefore it really is phenotypically significant. Yours might be the second hit, in fact, and then it might be significant. Going back one, this is what the overlap between the two biggest ones looks like now. ClinVar now has twice as many as everybody combined had back then. ClinVar is growing as people put more stuff into it. HGMD is growing more slowly because it curates the literature and gets them out of the literature. Nevertheless, there are a lot of them in HGMD that ClinVar won't have because people are not going back and putting their old ones in. So HGMD is still useful for what it's good for. And the version we have on UCSC is their public version, not the $4,000 a year subscription version. I tried to talk them out of it, but that's their business model, is to sell it to you and not let me show it to you. And I swore we weren't going to let anybody download it. We would just show you the tick marks, but they wouldn't have it. So we're 3 years behind their subscription model. Okay, so I was thrilled yesterday to see, whoops, it's cut off at the bottom of the slide here. There's an acknowledgement down there, the source of the slide. I should at least acknowledge him once there. Let's grab that and drag it up a little bit for you. There we go. In yesterday's talk, that a genome browser screenshot showed up while I was watching, and it shows you a couple of the data types. These are data sets that were loaded into the browser in Wiggle format, and so you can see that it shows you a peak in 2 dimensions. And these look like these are data that were loaded by the user, by Tomi or somebody else, into the browser. So we have a lot of data that look like this from the ENCODE project and the Blueprint Genomics project and a few others. But you can load your own data as well. And we're always thrilled to see slides at meetings and talks and so forth, because I used to be a molecular biologist, but I don't do much of that anymore. And so instead of publishing in molecular biology, I get my thrills when you publish something using our tools. And so it's always nice to see slides at meetings. So I want to thank him for using a browser shot. So here's another browser shot that he used yesterday. It's kind of squirrely in shape because I was sitting over there and I took the picture at an angle. And here's one that's all fuzzy because I took it in a hurry. But multiple tracks, you can still see here that there's a peak in this one and not a peak in those if you're looking at epigenomic data and so forth as wiggle tracks in the browser. And these are just regular gene set. It makes me dizzy looking at them, so I'll move on. And here's one that is not a genome browser shot, but it shows the number of individual variants at these particular locations. But he might not have known it when he made this slide, but just about a month ago, we released a new data format we call Lollipop, which is in a TCGA gene set that's been released on HG38 just about a month ago, maybe even a little bit less, which shows you how many times that particular variant has been seen in that data set. And so this is a cancer-relevant gene, mTOR, in the TCGA, the cancer data set there. And these particular variants have been seen 10 times. These other variants have been seen smaller number of times. And so this particular data format is available on the browser in this gene track, and it won't be long before it's available to you to load your own data sets in that format. It's not available yet in a user format, but it will be very soon. Kate's working on that right now. Or is it late enough in the day? Yeah, she might be working on it now. Okay, and here's another browser shot on this side of the slide. But right here, this reminded me to show you that we're working on a data format For that, Jonathan Casper in our group is working on a data format that would allow you to load data, to visualize data, high density, high C data in that format. And that'll be released within the next few months, I believe. So you can see this kind of data in the browser, in resident data sets, as well as loading your own data. And a little further out is our display. It's in our grant year, which doesn't end until June 30th. So sometime in the next 10 months, will be a way to load your own data as trios, so you can see the mother, the father, and the child together. And you can see here, this is a little fuzzy because it's like a screenshot of a screenshot. But you can see this upper chromosome here came from the mother. There's a little deletion there. And those two variants there came from the mother. But this variant came from the other chromosome. So maybe there was a rearrangement between them in the egg. Whereas this other chromosome came from the father, but that variant there is de novo. It didn't come from either parent, even though it's in the child. So you'll be able to see, if you have phased data, you'll be able to see the mother and the father and the de novo. If you don't have phased, then these two collapse and the variants come together, and you lose the ability to know which regions are linked to each other, because the variants come out of short reads technology unlinked. The reads are too small. If the variants are too far apart, they're on separate reads, and you don't know if it's this chromosome or that chromosome. You can't link them together without some long read massaging of the data. And so we're going to have that data type available as well. And so this is a paper that many of you are familiar with, I imagine, is the ACMG standards for interpreting a variant of your own that hasn't been seen before. And it helps you, guide you through the best practices, and it's used all over the world, even though it came out of an American effort, the ACMG. And what they recommend in their table 1 is go to all of these different places to get your data. And I'm going to go down the list kind of quickly. I try not to speak too fast because I don't want to get out of breath. I can actually get out of breath listening to someone else speak too fast. But the Exome Aggregation Consortium, the EXAC, we have that on the browser. 1000 Genomes Project, we have that. The EVS from Seattle, we have that. DBSNP, I showed you a slide, we have that. We have DBVAR, we have ClinVar, we have OMIM. We have the old HGMD, so we're not up to date on HGMD. We have HGVS, we have LOVD from Leiden, we have Decipher. Well, you get the picture. If you use the UCSC Genome browser as a background, you can get access to all of these different data sets. Down below is just the sequence themselves, and that's the backbone of the browser. So you can turn on the data sets on the Genome browser and then access and go out and read the data themselves, using us as a gateway to the other data. And here's a little cartoon showing those data in a matrix, large and small, and pathogenic and benign, and the different data sets that are relevant in these different contexts. And the second URL on your little mini handout has a link to a page called databases.html, which we can use to access saved sessions that will turn on each one of these 4 quadrants individually. Sometimes you're looking at big stuff and you want to see the databases that are relevant for that. And so you're interested in Decipher on the pathogenic side and maybe ClinGen and ClinVar. Or on the benign side, you might look at HGMD and Uniprot and OMIM, because they have some big stuff as well. And DBSNP has, I'm sorry, if you're looking at big stuff, you're also looking up here at DGV. You're looking at the benign big ones. And we have the gold standard now, which pulls out some of the older DGV entries that were not as reliable as the more recent one. And so I want to show you later how to use sessions as a way to save your train of thought. When you're thinking about this kind of data, you can pop right directly into a session that has those on, and then navigate to the gene you're interested in. And you don't have to remember where to find all of the different data sets. You can save your thinking as you go. So data, and I did not pull the slide out just because I'm in Kansas and Missouri here. But the paradigm for data is, we have data and we're going to mine it because we're going to publish it. And Furthermore, our IRB says we can't make it public and we can't share it. And that gives us a really good excuse to do what we were going to do anyway, is not share it. But as was said yesterday a couple of times, that second hit is pretty important, and having multiple hits is really important. And if you have a variant that's relevant but only present in the population at 1 in 20 or 30,000 in the population, which is kind of common for a rare variant, you're going to need 2 or 3 or 4 times that many patients before you see your second hit if you do the stochastics on it. And not all hospitals see that many patients, short of several years, before you see 50,000 patients. But there are a bunch of other hospitals out there that might have seen it. So how do you get access to each other's data instead of having them siloed and therefore not available to each other? There is a method, it's called Beacon, and a Beacon is a software program that's small and easily available, downloadable from the Beacon network that you can put on top of your own database. And I encourage you, if you have access to a database, to think really hard about doing this and sharing your data with the rest of the world. Somebody might call you up and say, I've got that variant too. Let's talk. What's your phenotype? Let's collaborate. Let's solve this case, the most important thing. And incidentally, maybe we can publish it because I've got this variant, it's a suspect. The beaconnetwork.org has software written in C and Python and Perl and a bunch of other languages that is not too hard to implement for a typical programmer. And what it does is it answers the question, do you have my variant? And most IRBs are okay with you saying, yes, I have seen that variant somewhere in my mass of data. Or no, I haven't seen it. And so that's all you're doing when you put a beacon on there, is answering the question yes or no. And it's even so specific that if somebody asks for a G to A transition, and if you have the G to C transversion, you say no. Sorry, I haven't seen G to A there. You know, so it's very minimal. And then if they are interested and you do have it, they contact you and you do a data use agreement and you swear to confidentiality rules and so forth, and then you can talk as collaborators, and it's not being exposed to the whole world. And the Beacon network has a network of, when I made this slide, it was 44. Last time I looked, it was 54 different beacons that are out there. And you can go to beaconnetwork.org, put in your variant, and it'll search all of those beacons and come back with, in this case, I found one out of the 44 said yes. And this was the Caviar database at ISB in Seattle. And so if I was interested then in talking to somebody about the variant, I go, that's great. One person's got it, let's talk. And that way you can get out of the siloing of data and, you know, little baby steps towards sharing data. And so all of these other databases have been contributed by consortia and millions of dollars of taxpayer money and thousands or millions of years of PI work and grad student work and sweat. And so we are now benefiting from all of that. Our way of giving back is to make our data available to the world. So I greatly encourage you to get to Beacon Network and figure out how to make your data available and share it. Okay, that's soapbox number 1. Okay, another thing, this is not directly relevant to the Genome browser, but it's brought up by one of my earlier slides. So I've got 2 versions of these data. These are the data from 2019 that I showed on a slide, ClinVar only, HGMD only, and the intersection. And then here's a slide that shows them an intersection. So I'd like a little show of hands. How many of you like the figure better? And how many of you like the table better? Most of you don't care. Okay, I want to get at least a third of you to answer. Table? Figure. Ah, okay, a lot of people like the figure. I don't like the figure. Because this figure is not proportional, and to understand the figure, you have to unsee the figure to read the numbers and then build a new figure in your head, which is way too much overhead for me. If it's a 3-circle figure, it's even worse. And so I would prefer to have it that way, and I can see for myself. This figure I do like better because it's proportional. So if you're making posters and figures, I highly recommend that you make your, have pity on your users or your viewers and do a proportional Venn diagram. There are proportional Venn diagram drawers all over the web. You can type in A, B, and C, and A plus C, and A plus B, and then get the figure, pick your own colors and so forth. And so that's just another soapbox. It's got nothing to do with the browser, but except that we're into data display, right? That's what we do, data display. And so this is an example of data display where you can do your viewers a favor. And it's the kind of thing we think about when we're engineering the browser, try to make things useful to you and intuitive. If you find something on the browser that's not intuitive, we have a mailing list. Let us know. We'd love to hear your thoughts. Y'all are way more numerous and way smarter than we are, and you have ideas that we haven't seen. So we'd love to hear them. And a lot of things that are in the browser came about just that way. Somebody says, you know, it'd be really cool if you did that, such and such. And you go, God, you're right, let's do it. And so we did. Okay, so I want to switch now to the live demo of the browser, and that's it for the static slide portion of today. And so I encourage you now to go to genome.ucsc.edu, and I will go over here, and I'll just go home to that place. I also want to encourage you to feel free to walk out and sate your thirst outside if you wish, to come and go. It's no problem to me if you need to get up, use the restroom or whatever. We may take a 10-minute break in maybe half an hour's time anyway, just to give us all a chance to stretch. So here's our home page. It's not very informative because all of these links are available inside the browser, and most people who use the browser link to the next page anyway. But the one place that is a little bit informative is our news feed down here. You may not care about the horse or the rhesus macaque reference assemblies, but you might be interested in the new data types when they come out, and this is where we announce it. And there are a lot of features that came out here on the browser that I will present in a workshop somewhere. I gave a 2-day workshop at UC Davis a couple of years ago, and a woman, a PI, came up to me at 10:30 at the 90-minute break and said, how long has that saved sessions feature been there? And I said, I don't know, 3 or 4 years. She goes, oh my God, it's already worth 2 days of my time. Because we've been emailing screenshots around. And you don't have to email screenshots around. You can just keep a link, or you can have a shared login where you can all see each other's thoughts with the saved session feature. We announce new features there. You can subscribe to us. You can see it's not very frequent, July, August, September, one a month. Sometimes as many as 3 or 4 a month at the most. So you can subscribe there and get the latest news. Because I know you all need more email in your inbox in the morning, right? There's no login required for that. You just subscribe, so no new password at least. So here's the Genome browser link. Let's click to that and go to the gateway where you have the option to choose from all of the different animals that we support. We're in an evolutionary tree there because we're very big on pushing back the borders of ignorance on that whole creation thing that seems to be really popular in this country. So we lean really heavily on the side of comparative genomics and the evolutionary story that's present in the genomes of the existing animals as well. And the default, which you get to, if you will, please go to Reset All User Settings. And that sets us at the default Human Genome Assembly HG38, the most recent one. You can see we keep the older ones. We used to archive the older ones. But it's actually more trouble than it's worth to archive them, so we just leave them up there. And we do get a small amount of traffic still on 17 and 18, and right now we're about 50-50 on 19 and 38, or 60-40 really. People are still using 19 a little bit more for reasons I'll go into in just a moment. One of the things about HG38 that is useful is if we go to the View Sequences menu here, you can see that these are the chromosomes. Each one of our assemblies has a link right there. These are the main chromosomes, but we also have a bunch of alts and fixes. And the alts and fixes on chromosome 1 go down to there and below. Those are places where the consortium has made some changes in the reference, or the alts were in the original reference, but it changed. There were places that are fairly variable in the human population, and so they gave you alternate versions of particular chunks of the chromosome. You can see they aren't very big. On chromosome 6, there are a few that are 5 megabases, but most of them are under a megabase. But the the MHC and HLA regions that are immunologically relevant are highly variable. And so they've been in the genome for a while as alt sequences. So some of your reads may map to these alts. And so your bioinformaticians need to know about those if they're going to use HT38, because if something maps 4 places, it might map to 3 alts. So it's really uniquely mapping, not 4 places in the genome, but 4 copies of the same place in the genome that have been rearranged. And those are different things. And so your bioinformaticians need to know about that. And that's part of the reason why HT38 is not as widely used yet, because not everyone has written pipelines that are sensitive to that. And the reads that are multiply mapping are thrown in the bin, or they're using HT38 with just the main chromosomes, which means they're getting the advantage of some of the new stuff, but not all of it, because they're ignoring the alts. And so some of your reads may be going in the alt. If you have people whose ethnicity, did I mention that RP11 was from an African-American guy from Buffalo, New York? Which means he's got the most variation of, that population has more variation in an individual than people from Western Europe and Asia, because of the out of Africa thing was relatively recent. And so if you have people who are not African in descent, you might consider asking your bioinformaticians to map to the alts, just see what kind of grumbling you get out of that. So I'm going to go back to the browser here. Whoops, I didn't mean to click into there. I wanted to go back to the genome first. Click that link and switch to HG19. And another reason why people are using 19 more than 38 is that there are a lot of data on 19 that haven't been lifted to 38. And so nobody likes to jump back and forth. I'll show you an easy way to jump back and forth between them, because there are some people starting to map to 38, but most people are acknowledging that fact and mapping to both. The ENCODE project and Blueprint Genomics, epigenomics projects are on 19 and not on 38. And it would take $1 million to hire people to do it. There's no postdocs going to do it, because there's not really a job in that. That's just running a script on things and lifting data. Okay, so let's go to HD19, click in. And here's the default location and the default tracks turned on. So the first thing you notice is it's kind of busy. We found a place with a gene that has only a single isoform there, the SOD1, for the default location, just so you'd see something. The browser shows you a little block wherever the coordinate lines up with a thing. Here's a peak that shows you where this particular histone modification, this acetylation on K27 is. It's near the end of the gene right here. Multiple cell lines there in different colors. That's a wiggle track that shows you that, and it maps right to those locations. This box is an exon. This narrow box is an exon also, but it's untranslated, so you can see where the start and stop codon is that way. But the reason I belabored that point, that things line up with the coordinates, is this one track doesn't. The GTEx track, which shows you 53 tissues, it shows you the gene expression for the entire gene. So what's lining up there is this bar along the bottom there. It shows you this whole gene has been normalized, and all of these different tissues are being analyzed in this project, and so you can see each of the tissues. This particular gene is expressed relatively uniformly in most tissues. It's a little higher in, this is a log transform, it's a little higher in liver than in other tissues. And so if I put my mouse on the coordinates system above the browser graphic here and drag just to the right with that exon in the middle of it, I'm given a menu here which lets me zoom in. Before I actually zoom in, though, I want to move it out of the way. And so, okay, so what it's going to do is take this whole strip here and expand it. But what's going to be a little bit of a surprise is this graph is going to show up because we crossed this line. And so it's not going to just expand the line, it's going to redraw the graph. So that's this GTEx track is a little weird in that it does that. All of the other tracks just get fatter when you zoom in like that. So if I hit zoom in there, it's now just a little wider, and we're looking at that exon. We're zoomed in on that exon, and we once again have a graph of the entire gene's expression in multiple tissues in the body. Before I turn that track off, I want to click into this little button next to the browser, or next to that track. Before I do that, I want to ask how many of you have used excuse me, the UCSC Genome Browser a little bit. Okay, a few of you have used it a little bit. Of those who have used it, how many of you have clicked into a little button like this next to one of the data tracks? Not everybody, but a few. This is the best kept secret of the Genome Browser and one of the most useful. It's a configuration button that every track has this little button. And sometimes you can turn on more data, that there are other data sets that are hidden. Other times you can get configuration options like change the height of the track and so forth. And in the GTEx track, you get a lot of control because you don't have to see all 53 tissues if you don't want. You can use the body map over here and, for example, turn off the thyroid if you don't care about the thyroid, and turn off the maxillary gland and amygdala and so forth, or turn everything off and turn on just adipose, and then go back to the browser like that. And now we have a single bar, a single value, so you could just look at a couple of tissues and so forth. And so this is what we call the bar chart data format. And it's actually available to you to load your own data into the browser if you have 4 or 5 cell lines or 4 or 5 different patients, or tumor versus normal. You just have 2 different kinds. You can load them into the browser and see peaks to see, or if you have 5 treatments, no treatment, or 0, 1, 2, 5, and 10 micromolar of some drug, and you can see how your cell lines are going up or going down with time. You can condense all of the data into a bar chart format like this, and you can load your own data in like that. And so I will use my right mouse button to the left side of the track here and hide that particular data track, and draw your attention back to the gene track here at the top. You can see we've got amino acids in there, just barely enough room to draw the amino acids in there. I see one person squinting at least, so I'm going to show you the configure button here. Make the type a little tall, make a type a little bigger by hitting that configure button. I'll move it from 8 to 10 and hit submit. Okay, now I've made the type bigger, but it doesn't fit in the boxes anymore. And so we show codons by alternating stripes of light and dark. Try to give you a sense of scale. Otherwise, you got to go up to the top here, subtract 2 numbers, divide by 3 to figure out how many codons you're looking at. That's a bit of a hassle. So we try to give you as much information on screen as we can. Here's where we are on the chromosome itself, a little tick mark on the right arm of chromosome 21. If I zoom into just a small number of amino acids, you see that we actually have room to draw the number, and you can see that this particular glycine is amino acid something 34 with my glasses on. And so you can see, if you're zoomed in, you can see which way is it transcribing, right or left, and so forth. If you are really annoyed, when I first joined the project, I was such a molecular biologist that having genes on the wrong strand going 3' to 5' just drove me nuts. And so I complained about it bitterly until Mark Decons, one of our engineers, unbeknownst to me, he actually one day showed up with the reverse button. And he said, "All right, Bob, shut the hell up. Here's the reverse button. You can flip right to left now, and you can have your genes on the 5' end if that's the way you want it. So if you're looking at the same gene all the time and you really prefer that, you can flip it." He had the wisdom to put this text box over here on the right so you can tell it's backwards so that you're not confused. He even flipped the ideogram so you can see here the centromere is right of center. The paradigm is P telomere, P arm, centromere, starting at coordinate 0. And so this is flipped, so it puts the P arm on the right. So your acrocentric chromosomes are going to have the centromere over here at the left arm unless it's backward. Is that a question?
Yeah. You have the arrows. What's the direction of the transcription bigger?
Can you have the arrows bigger? You can. I think by increasing the font, you get bigger arrows also. Everything gets bigger then, too, so you get a little bit of less data on there. Speaking of arrows, this little arrow right here, which let me switch back. Let me reverse it back here. I got over it finally. I can read backwards now without freaking out. But so anyway, that gives you a hint about my background. My background is in molecular biology, not bioinformatics. This little arrow right here reads the DNA left to right, and you can see we have GGTG. If you flip it, then you reverse complements. And so now we've got CCAC there, so you're seeing the opposite strand. And so if you wanted to look at the actual DNA of a gene that was on the other strand, you could leave it in reverse and flip the coordinates as well. So you get this arrow if you're zoomed far enough in such that the nucleotides are visible in the top of the window there. The multi-species alignment here, you can see here that the conservation of 2 nucleotides, but not the 3rd nucleotide because of the wobble base, is very graphically obvious here that GG anything gives you a glycine. And so you've got a strong signal where the G's are. And even though all the way back to Xenopus, you have a G at this amino acid, you have wobble at the 3rd base. And so the DNA tends to decay away in the 3rd base. Let's click into this real quick, into this, and turn on all of the species. Click into the configuration button, turn on all of the species, and then submit. Did I miss the button? There it is. Hit submit. And so now we have all 100 species, and you can see how well conserved this amino acid is all the way down. That G's there all the way down into the fish, lizards and fish and so forth. Looks like zebrafish has no sequence in that particular location. So you can see the entire 100 species if you wish. We don't have them all turned on by default. Using the right mouse button, I'm going to, before I do, let's zoom out by a factor of 100. I want to show you something that's kind of interesting about the signal. So here's an exon in the middle of the page. Okay, so how many of you kind of lost where I was? I clicked and I clicked and I clicked and I moved around and you got lost and you got lost. Okay, I'm going to save a session so you can all catch up. And you can get exactly this picture, and I'll show you the value of saving sessions. So I'm going to go view, my data, my sessions. And I'm going to sign out and then sign back in as username example. And just to show you that I can save a session and you don't have to be me to see it. So I'm going to save it. I'm going to call it HD19 underscore KC Mercy 2, because I saved another one called KC Mercy 1 already. And so now, if you want, you can go into this interface right here. And if you know my name, which is example, so you can type username example right there, and session name HG19 underscore KC Mercy 2, and hit submit, then your page should look just like my page after you hit the browser link. I'll give you a second to catch up there. KC Mercy 2. I always keep the prompt so I don't accidentally go to HD38 when I click on one of my sessions, or mouse, God forbid, and I'm at mouse and don't even realize it. And so I keep the prompt that tells me which genome assembly I'm using. And so if I click on the browser link up here, okay, I have a little buzzing. Did people find it that wanted to?
Do we have to sign in?
You do not have to sign in. You just have to know that when you restore a session, my username was example, and my session name was HG19 underscore KC Mercy with a capital M 2. It is case sensitive. And so that feature lets you save a browser view with anybody that you want to share it with. I'll go back to my data, my sessions, and I'll reload this session here, and I'll show you that loaded sessions. When you first save a session, you also get a link here that you can share with somebody else, or you can use the right mouse button and get a slightly different version of the link by copying the link address there. And so you can save that, and we save it as long as we feasibly can. We do occasionally have a hardware or software problem. We've lost data in the past, much to our chagrin and our efforts to back things up. So I will leave it up there until I see people are comfortable in having found the page. So the session is named HG19 underscore KC Mercy 2 with a capital M. After that, you hit submit, and then you click this browser link, and it should go back to the browser. Did it go loaded settings? Did you see updated settings? That little place, when you first do this, that's kind of cryptic. And I've argued internally to put this in red or flashing or something the first time. So it's a little bit more obvious. But I haven't quite gotten the attention of the engineers to do that. We don't do flashing anywhere. I knew they would never do that. So did you get it? So it's worth, yes?
Is there like an undo button on this, where if you do something and you just want to go back to what you were doing immediately before, you can hit undo?
I think the back button should help you do that. In general, we don't encourage the use of the back button, but the settings thing is an undo button itself if you want, because you can save something, then you can go away and you can come back to where you were, use it as a bookmark. Yes? The session name lasts as long as we can. We still have sessions from 2005 or something when we started this. We have lost some data. I don't think we've lost any sessions. We've lost some custom data that people saved with their sessions. We had a problem with one of our servers and one of our backup servers at the same time, and we lost some data. We spent a bucket load of money to get a lot of it back. But yes. It'll create a new URL every time you save a session. And I don't know if you can see on mine, I got 271 settings saved under my name. And I've got a bunch of them saved under other names because I've created a login for you all. As long as we're here and you want to log in, I'm going to sign out. And I created a login to share, which is easier than having you say, each of you do it, because then you have to go to your email. You have to create a password, go to your email, authenticate that you're not a robot, and that's a pain. So username, KC Mercy, 2019. Password Genome. And then you can log in, unless I got it wrong. Genome. I got fat fingers. Maybe I called it Kansas City. I forget. I could go check my email, but I probably deleted that one. Kansas City 2019 Genome. There it is. Kansas City 2019. Capital C. Yes?
So another question. So say you're the one who made the session, and then you shared it with someone else, but then 5 years later you decide you don't want that anymore and you delete it. Can the other person, like, do they need to save it when they get it?
Right. Yeah, the question is, if somebody else visits my session, what happens when I delete it? That link won't work anymore. So if somebody sends you a session and you really like it, save it yourself, and then you have a new name for it. Because it's the starting point for going out, but it lives as long as the user that built it keeps it. And so you're welcome to create your own yourself, but you have to have a password, and then you have to, if you're not going to do that long term, no point in it. You can use this one. We've got a lot of sessions that I have saved at workshops through the years, and they're still there. And so if somebody wants to create a session right now, they could do that. And actually, let me reload. Nobody has it yet. I can save the first one, HG19 _ KC Mercy 2. I'll save it there. And so now we have one session saved. And so if you're logged in as Kansas City 2019, you can get there just by clicking this. Go directly there. You don't have to use that reload interface at all. You just simply click it, and there it is. So did everybody get to the session that wanted to? It was worth stopping because it's a feature that's so useful that once you've played with the browser a little bit and you have something you want to hang on to, that's the way you do it. You're welcome to use Kansas City 2019 as long as you like. But beware, other people might overwrite your sessions because if it's a shared login. So if you scroll down the page here in this conservation track, you see that as time goes by, evolutionary time, because these are in order, first the great apes, then the non-ape primates, and then the non-primate mammals down the list. As you go down the evolutionary tree, the signal starts to decay away until you're down here among the among the turkeys and fish and so forth. The only sequence that's left, well, there's a chunk over here that's left, but the main thing that's left is the exons. And that's because the gene is conserved, and there's been selection at the amino acid level for the sequence of the protein, and the introns have decayed away. There is a DNase hypersensitivity site over here, which kind of, whoops, doesn't line up with this other sequence. So you might be curious, whoops, I clicked into it by accident. So we have to wait for it to load. It's going to show us the details of that, of the sequence of all the different species, which I had not planned to do, to show you. I'll just click back to the browser from there. And so another important feature of the browser is getting around in the browser and knowing what's in there. The SOD1 gene, like all of our other genes, if you click into this gene, you get this matrix of data that accesses data sets all over the world. It tells you what SOD1 is, superoxide dismutase. It tells you biochemically that it binds copper and zinc. It's a soluble cytoplasmic protein. It's a homodimer, and it's one of the causes of ALS. And so you can get a snapshot at all kinds of levels, at the enzymatic level, the biochemical level, the genetic level, and the phenotypic medical level. This comes directly from the RefSeq record at NCBI. You also have a lot of other data on the page, links to pathways. If you click the pathway link here, it'll go down the page. Oops, or it should. There we go. We have imported data from 4 different pathway databases, and you can click into the Huntington's disease pathway at Kyoto, Kyoto Encyclopedia of Genes and Genomes, and see the other genes that are associated with it in the pathway, and it will be highlighted in red. I have not figured out why this other gene is also highlighted in red there. It's red type, but not a red box. So I haven't looked into why HTT. I have clicked into it before, but I don't know why that is. And then if you just crash that window, you're still on the browser. So we have a lot of data on the Genome browser that gets you access to other locations, and we always open up a new window so you don't lose track of where you were on the browser. Scrolling up the page here, we have gene ontology data. It was mentioned in a talk yesterday also, at the cellular level, at the molecular level, and the biological level. We have links through to model organisms. We have crystal structure information. And for example, and if you click into Muppet here, which is a new feature that we just released, comes out of Johns Hopkins, you can see the protein if the crystal is known. And I think I don't know, I don't see the zincs in that one. Sometimes they have the zinc and copper ions in there. This one here also gives you a 3D image, but we're still one click away. Whoops, it's removed from distribution. Okay, maybe not that particular link. And here's a repeat of the GTEx data, but this is in a linear scale instead of a log scale. And toxigenomics information, small molecules that interact with this gene, or the gene product, I should say. A little bit about GWAS studies that are known for the gene. And Unipro, yes, please. I'm sorry? How did I get to this page? The structure was just above the pathways link, and it's protein structure link in the box there. So this upper box is a page index, so all that stuff is already on the page. So you could find it by scrolling the page or by clicking there. It's a tall page though, so we give you access more quickly that other way. The stuff in blue down here, you can also get the RNA itself. And if you clicked into a particular isoform, which you would have, you get the RNA of that isoform and the amino acids that are encoded by that particular isoform of the gene. SOD1 has only one, so you only get one. There's only one choice right there. You can also get the genomic sequence from this region. These other links go to other places. You can click through to OMIM right there. You can click through to our friends at Ensemble. the Human Genome Nomenclature Commission and so forth. These links, the mouse people at Jackson Lab, what's known about this gene, all the different mice mutants that are available, the different variants that are available. And speaking of mice, I probably won't remember to show you later, we have a track on there that it's called the Knockout Mouse Consortium that shows you all of the different available cell lines and frozen embryos and so forth for which a knockout mouse is available. We have it transmapped onto human so you can see the human gene. You can click through and you go, oh my God, I could find a mouse that's missing this gene. If the gene, just a moment, if the gene is lethal, they make a constitutive lethal out of it by putting it between a Cre-Lox cassette so that you can grow it up and then knock it out at will later on. Yes, your question. Oh, the knockout mouse? It's the entire consortium, right. There is a problem with getting some of those reagents into the country, though. At the border, they look at this and it says mutant mouse and freak out. And then they sit on the loading dock while they figure out what to do with them, and it dies. So I have heard people that tried to get reagents, if you will, live material from Germany, and never got it. But they are available some way. Maybe you get DNA to transform it yourself or something like that, transfect it yourself. So that's the matrix that the known gene details page gives you for each of your genes. And just for fun, let's go into the mRNA here and grab maybe, I don't know, 30 bases of that RNA and copy it. I'll just highlight it and copy it. And then I'll go back to that this page and then go forward to the genome browser. And before I use what I just copied, let's use the right mouse button over here on this on the little configuration button and hide all the comparative genomic stuff. And I'm going to paste the DNA into the box, into the position box. This is a relatively recent feature in the browser that we now support BLAT right there in the position box. So if you, we got one hit, you can either see the details of the match. It's going to be a perfect match because we know where we got it. So I won't do that, but I'll click into the browser link. And where to go? My sequence. Oh, it's spanned an intron. Look at that. So it found the sequence. Let me zoom out a little bit. Zoom out by 10x. And so there's my hit. There's the DNA I used, or the RNA I used. So if it's long enough, it needs to be 20 bases or more to align to the genome using BLAT. You can find your location really quickly anywhere in the genome. It does not work with amino acid strings because no matter how, sorry? Yeah, I simply pasted where I got the DNA in the first place. Okay, I was in the SOD1 record. I clicked to RNA. Back. Okay, I'll go back one. And thanks for doing that. Thanks for slowing me down. There we go. Okay. Thank you so much for slowing me down. I really appreciate that. So I clicked into the gene. Got it? Got that? I clicked RNA right here at the top of the second position box, or the second navigation box. The mRNA that supports this gene annotation, here's the RNA itself. I grabbed a chunk of RNA out of there and then did, at random, I did Control-C. I'm glad I made it long enough so that, because it hit in two places, BLAT's not so happy when you have two small pieces that are separated by an intron. And so then I copied that, and then I went back.
You're using the back button.
I did use the back button to get from that text page back here to this page. And then I used forward navigation on the genome browser link up here just to go back to the genome browser where we were before. And so here we are at the same page. Then I pasted the RNA right in there. It's actually DNA, right? It's ICTs in there. So it said mRNA, but it's giving it to you in DNA. And then when I hit go, it found it. So this position box is good for a lot of things. For example, you can type in, what, ASCL and choose. I think it was 1 we were looking at yesterday, one of the genes that came up in one of the talks yesterday. You can choose it from the drop-down menu and hit go. So you can navigate straight to the gene. While I'm here, I'm going to hit the hide all button and turn off all the genes, turn off all the data tracks rather, and just turn the genes track back on to pack. Oh, actually, I'm not even going to turn it back on because it'll come on when I use another gene. So I did hide all, and then type in, I don't know, EGFR. And if that's actually a gene name, you choose that and hit go. It turns the gene on. You can also do something like FGFR2 space P dot 44. And if it has 44 amino acids, it should take you to the 44th amino acid. No, please. Please say, oh, cool. I love that. There's a one caveat here that it only takes you to the one of the what we call the canonical isoform, the isoform that has the highest rating with RefSeq. So if the higher the number, the more chance you might be in the wrong isoform relative to what you're looking for, because the numbering starts at the methionine, and when you throw in another exon, the numbering gets out of phase. So the two isoforms have different numberings. So it might not be the isoform you're looking for. This particular one right here, there's an isoleucine in the same location that's called 63 in that other isoform. So that tells you right away there might be a different start site or an included exon right there.
So is the top one the canonical isoform?
Not necessarily, because they're ordered by leftmost start site in the bioinformatics sense. It could be the stop site on one like this, which is a minus strand, by the leftmost coordinate. The way to find out what the canonical one is going to be is if you type in FGFR2, it will come up highlighted. Whoa, is it broken? It might be broken. It's supposed to be highlighted.
All right, so in theory, when you,
It won't necessarily be the top one.
okay.
Right.
But if you do it like that, it'll highlight.
It should highlight it. In fact, let me make a note here. Did other people have it highlighted? Yes. Okay, so what kind of browser am I using here? I'm using Chrome. Okay, that's good to remember. If you do write to us with a bug, it doesn't hurt to tell us what genome assembly you're on and what browser you're using, because we're going to have to write back and ask that, probably. One thing you can do to identify which isoform is you can turn off all the other isoforms. So if you click this little box on the left here, you can turn off the splice variants. And then if you go back, the one that's left is the canonical isoform. That's the one. If you want, you can highlight, let's see, highlight FGFR2. It should highlight just this one. And then if you zoom out a little bit, you've got just this one highlighted. So if there are different start and end sites, now we'll turn back on the splice variants. And it may help you narrow it down to 5 of them. So it's one of those 5. It was the canonical one. So that's not it's not trivial, but it does help a little bit. That's all I can say. You can see here we have the other gene set here turned on. The NCBI RefSeq gene came on when we did the P dot, because the HGVS nomenclature is keyed to NCBI RefSeqs. And so you can type in NM underscore, you know, the standard HGVS, NM underscore number colon. That we support a lot of that kind of navigation. In fact, I'm going to open up a fresh browser window, and I'm going to go to that second URL I gave you on your little mini handout, bit.ly/KC Mercy 2019. And we have a little file down here called hgvs.txt. If you click into that, you can see some of the formats that we support, and we can actually copy them over onto the browser, do a little copy paste. So we already showed you this one, right? Where you just put, you don't even need the dot, I guess. Just P141 works. Because I did P dot a moment ago when I showed you that one. Let's just grab the top one, UBE3A. You'll notice that it says that it's case sensitive. So you have to have a capital G and a capital T for the glue and the threonine there. Let's go back to the browser, paste that into the position box. And just for fun, I'm going to change it from threonine to tyrosine to show you what happens. Oh, actually, I guess nothing's going to happen because it doesn't really care what it's changing to. So if I change the glue, it would complain. So then we go. And so here we are at the 130th codon of one of the isoforms of that gene. You can also see that it's got other numbers in other isoforms. We're working on, as soon as Angie gets finished with her current project, she's going to work on an algorithm that will, let's say you typed in E107, it would go to 107, but it would go down, it would go to the other isoform. It would go to the isoform, the canonical isoform. So what she's going to do is have it look through all of the 107s and see which one might be a glue and go to the right one. That takes a little bit more sophistication because some of these have multiple things there. Other things you can type into the browser, you can type in, before I do that, let's turn off the RefSeq gene set. And you can type in 6q13. If chromosome 6 has a band named Q13, it will go there and it'll show you in the little icon up here where you are on the chromosome, and it'll show you all of the genes. Now, if you have a deletion, a microdeletion or something of a large region like this, you may not be interested in all the isoforms of the AIMS1 gene and so forth because all of the isoforms are deleted or duplicated. And so that's the time when you might care about hitting the splice variants button over here and turning off the extra isoforms. It cleans up your screen and shows you just the genes that are just one copy of each gene. The more you zoom out, the dirtier the screen gets, the more busy it gets, and so you can do that. Once you're here, though, you might want to print out something like that. There's a number of different ways you can share this information. You can right-click it and just view the image itself. That's a PNG image, so you could copy-paste that into a slide or something like that. But if you wanted to put it into a publication, it would be fuzzy when you tried to, it's only 96 dots per inch, so you wouldn't necessarily get, Nature or Science resolution graphics. What you can do is you can go view PDF and export it as a vector graphic picture and download the current browser graphic in PDF. And now you have, let's see, if I open it in Acrobat, I can zoom. And so now you have a graphic that's scalable. And if you just want to look at this one region, you can scale it all the way up. And now you can save it at 300 dots per inch and have a big picture. So you can make it a column wide on Nature. And I'm really looking forward to seeing that, actually. So I found it under the View menu in the browser graphic. Do I want to save changes? No, I only zoomed. Let's go back here. I'll crash that extra window, and I'll go back to the Genome browser and show you that right here, View PDF. It was in the View menu. And so that's a good way to get your browser graphic out and make something that's cleaner and bigger and a little bit more viewable. Another thing you might want to do is you might want to see these two genes that are on right and left, but they're really far apart. but you might want to scrunch the screen down a little bit. If I make my browser window a little narrower, my Firefox window or my Chrome window, and hit resize, then the browser graphic will redraw with the new window size. And so that graphic now matches the size of the window there. And so you can scale it that way by squeezing it together, and it gets a little taller that way because all the same data are still being shown. And so that's another thing you can do to manipulate that, and changing the font size to play with your graphic before you have to resort to Photoshop. Just trying to give you a little bit of an idea of the kinds of things you can do. And if you really do like that, oh, I guess it's an RIMS1. If you like that rim gene right there, and you want to highlight that region, you have a choice of a lot of different colors. You can choose any color you like to highlight it. And so if you zoom out by, a factor of 10, a really dark highlight will still keep an eye on it, on that gene for you, and you can scroll down the page. Yes?
I wanted to just share a quick secret I learned. I don't know what you were going to say, but you can also do that by hitting the keys HM.
The keys HM. Highlight the window.
It'll highlight whatever you have your region that you
It highlights your region, right. That's cool. That's good. Thank you for that. That one, if you put your mouse over here in the blue region and type a question mark, you have access to keyboard shortcuts. And I don't know if HM is on there. HA. Clear all highlights. HC. There it is at the bottom. Highlight all. HM. It highlights the whole screen. So if there are things you do a lot, like zoom out 10x, or zoom to the base level, or something like that, you could learn a few. I typed a question mark. Not in the position box, but I clicked somewhere else on the screen, like, oh, I pointed to my screen. See right here? Sorry. Right, just anywhere over here. And then I typed a question mark on my keyboard, and it popped up this menu. My colleague Max says that a lot of websites do that. I said, really? I didn't know that. That if you type a question mark, you get keyboard shortcuts. The only one I ever use is default view. When I'm in Exxon only mode, I use default view to get out of Exxon only mode. So there's a handful of them. The J, K, L, and I keys down here you might recognize if you have played computer games in the old days. That's right, left, up, and down. Those are the 4 keys that you play with your right hand to move around on a screen. On some old video games. I'm going to right-click this bright red one here and remove that highlight. And I guess it defaults to the last color you chose, which I never knew before. So thanks for pointing that out. And I'm going to, let's make it a little fatter again. I'll scroll down here and I'll resize it to the screen size. Another thing you can do to navigate, and then we'll take a quick 5-minute break, is if you click here, you get this colon and hyphen and comma format to put in the browser. You can also copy paste from something like a spreadsheet or something else, where you can simply type in 6 and then 10 million space 11 million. And you don't need the punctuation at all. You don't need the colon, you don't need the hyphen. So it'll go to that particular million bases on chromosome 6. And so a lot of programs output stuff that doesn't have the CHR that Santa Cruz uses. It'll have just a 7, just a bare number for a chromosome. You can put that right into the genome browser now without having to put the CHR in there. Okay, so this is a good time to pause. Let's take a quick break. They only gave me 3 hours. Actually, I shouldn't say only. I thank you so much, actually, for giving me 3 hours. I'm really honored because most of the other talks are not 3 hours. So let's take 10 and come back at 36. Take 10 for a quick bio break.